Hi, this is Alex from devstories.eu. Scanpy Single Cell Analysis, Episode 8 of 16. The secret to modern single-cell analysis is not just plotting points on a grid. It is understanding exactly which cells are neighbors in a massive high-dimensional space. If you get that underlying structure wrong, every visualization that follows will mislead you. The mechanism for capturing this structure is the nearest neighbor graph, and today we are looking at how to build it and project it using UMAP. At this stage in a typical pipeline, you have already run principal component analysis. You have condensed thousands of genes down to maybe 40 or 50 principal components. But 50 dimensions is still impossible to visualize, and it does not explicitly tell us which cells belong to the same local community. We need to construct a web of connections. In Scanpy, you do this using the function sc.pp.neighbors. This step computes the neighborhood graph of your cells. Calculating distances between tens of thousands of cells across 30,000 raw genes is computationally brutal and highly susceptible to noise. By computing the neighbors on the principal components instead, the math is fast and the technical noise is already stripped away. For every single cell, the algorithm looks at its coordinates across those principal components and finds its closest peers. By default, it links a cell to its 15 nearest neighbors. The output is a mathematical network where cells are nodes and the edges between them represent high similarity. This neighborhood graph becomes the foundational data structure for downstream tasks. Once you have this web, you want to actually look at it. This is where UMAP comes in, called via sc.tl.umap. Here is the key insight. A very common misconception is that UMAP computes distances directly from your raw gene expression data. It does not. UMAP is entirely blind to your genes. It is simply a layout engine. Its only job is to take that pre-computed nearest neighbor graph and flatten it out into a two-dimensional space. UMAP works by optimizing a layout to match the graph. It pulls connected neighbors close together while pushing disconnected cells apart. Because it relies entirely on the local connections defined in the previous step, it is exceptionally good at preserving local structure. If a group of cells were tightly connected in the high-dimensional graph, they will form a distinct tight island on your 2D UMAP plot. But be aware that the empty space between the separate islands on a UMAP plot means very little. UMAP sacrifices global spatial accuracy to ensure local neighbors stay glued together. This separation of concerns is vital. The shape you see on a UMAP is completely dictated by the neighbor graph built right before it. If you want to change how sensitive your layout is to rare cell types, you don't tune UMAP. You go back and adjust the number of neighbors in the graph construction itself. If you find these episodes helpful and want to support the show, you can search for Dev Stories EU on Patreon. That's all for this one. Thanks for listening and keep building.